to
New notification.
finish reading this
Now
chapter.
broadcasting.
Turn this off.
Text in document. Share screen. Broadcast. Spectral.
The cell walls in plants and many microscopic algae derive their strength and rigidity from cellulose, along fibrous polysaccharide. Because of this role, cellulose is probably one of the most common organic substances on the earth. Interestingly, it is digestible only by certain bacteria, fungi, and protozoa. These microbes, called decomposers, play an essential role in breaking down and recycling plant materials. Structural polysaccharides can be conjugated, chemically bonded, to amino acids, nitrogen bases, lipids, or proteins. Agar, an indispensable polysaccharide in preparing solid culture media, is a natural component of certain seaweeds. It is a complex polymer of galactose and sulfur-containing carbohydrates. The exoskeletons of certain fungi contain chitin, a polymer of glucosamine, a sugar with an amino-functional group. Peptidoglycan is one special class of compounds in which polysaccharides, glycans, are linked to peptide fragments, a short chain of amino acids. This molecule provides the main source of structural support to the bacterial cell wall. The outer covering of gram-negative bacteria also contains lipopolysaccharide, a complex of lipid and polysaccharide responsible for symptoms such as fever and shock. See chapters 3 and 11. The outer surface of many cells has a sugar coating composed of polysaccharides bound in various ways to proteins. This combination is termed a glycoprotein. This structure, called the glycocalyx, serves as a protective outer layer, and also can play a role in attachment of the cells to other cells or surfaces. Small sugar molecules on cell surfaces also account for the differences in human blood types. Viruses also have glycoproteins on their surface with which they bind to and invade their host cells. Medical moment. Delivering essential nutrients. It is important to maintain homeostasis in ill patients. Supplemental nutrition is often necessary. For some patients, intake of calories by mouth or feeding tube is not possible. In order to supply essential minerals, vitamins, electrolytes, amino acids, glucose, and fluid, nutrition via intravenous IV line may be initiated. Total parenteral nutrition, TPN, is prescribed to meet the entirety of the patient's daily nutritional needs, accounting for their specific disease process and organ function. In addition, fatty acids might be prescribed alongside TPN because they provide essential energy stores and help with the absorption of some vitamins. This intravenous nutrition provides the body with energy patients need for maintenance of cellular processes and promotion of healing. Use context in the paragraph above to deduce what the word parenteral means. Answer. Page 19. Lipids, fats, phospholipids, steroids, and waxes. There are four main types of compounds classified as lipids: triglycerides, phospholipids, steroids, and waxes. The triglycerides are an important storage lipid. This category includes fats and oils. Triglycerides are composed of a single molecule of glycerol bound to 3 fatty acids. Figure 1.11. Glycerol is a 3-carbon alcohol with 3 O groups, and fatty acids are long-chain hydrocarbon molecules with a carboxyl group, CU, at one end that is free to bind to the glycerol. The hydrocarbon portion of a fatty acid can vary in length from 4 to 24 carbons, and, depending on the fat, it may be saturated or unsaturated. If all carbons in the chain are single bonded to 2 other carbons and 2 hydrogens, the fat is saturated. If there is at least one C equals C double bond in the chain, it is unsaturated. The structure of fatty acids is what gives fats and oils. Liquid fats. They're greasy, insoluble nature. In general, solid fats, such as butter, are more saturated, and liquid fats, such as oils, are more unsaturated. A two-part illustration of triglyceride is shown. Figure 1.11 synthesis and structure of a triglyceride O. Because a water molecule is released at each ester bond, this is an example of dehydration synthesis. The jagged lines and R symbol represent the hydrocarbon chains of the fatty acids, which are commonly very long. B structural and three-dimensional models of fatty acids and triglycerides. 1. A saturated fatty acid has long, straight chains that readily pack together and form solid fats. 2. An unsaturated fatty acid, here a polyunsaturated one with 3 double bonds, has a bend in the chain that prevents packing and produces oils. Right. Stock by punch stock. Access the text alternative for figure 1.11. In most cells, triglycerides are stored in long-term concentrated form as droplets or globules. when they are acted on by digestive enzymes called lipases. The fatty acids and glycerol are free to be used in metabolism. Fatty acids are a superior source of energy, yielding twice as much per gram as other storage molecules. Carbohydrates. Soaps are K plus or Na plus salts of fatty acids whose qualities make them excellent grease removers and cleaners. See Chapter 9. Membrane lipids. Phospholipids in membranes have a hydrophilic, water-loving region and a hydrophobic, water-fearing region. The hydrophilic region carries a negative charge due to a phosphate group attached to an alcohol group. The long fatty acid chains are uncharged and make that portion of the molecule hydrophobic. Figure 1.12. When exposed to an aqueous solution, the charged heads are attracted to the water phase, and the nonpolar tails are repelled from the water phase. Figure 1.12b. This property causes lipids to naturally form into single and double layers, bilayers. When two single layers of polar lipids come together to form a double layer, the outer hydrophilic face of each single layer will orient itself toward the solution, and the hydrophobic portions will become immersed in the core of the bilayer. This behavior allows phospholipids to be the main constituent of all cell membranes. A two-part illustration shows the structure of a polar lipid molecule and phospholipids. Figure 1.12 phospholipids. Membrane molecules of a model of a single molecule of a phospholipid. The phosphate alcohol head lends a charge to one end of the molecule. Its long, trailing hydrocarbon chain is uncharged. B. The behavior of phospholipids in water-based solutions causes them to become arranged. 1. In single layers called micelles, with the charged head oriented toward the water phase and the hydrophobic nonpolar tail buried away from the water phase. Or 2. In double-layered phospholipid systems with the hydrophobic tail sandwiched between two hydrophilic layers. Access the text alternative for figure 1.12. The oils on duck feathers keep these two canvasback ducks insulated and dry, no matter how much time they spend in the water. Oils on duck feathers keep these two canvasback ducks insulated and dry, no matter how much time they spend in the water. Copyright Guy Critchand and Getty Images. Page 20. Steroids and waxes. Steroids are complex ring compounds commonly found in cell membranes and as animal hormones. The best known of these is the sterol called cholesterol. Figure 1.13. A sterol is a steroid that has an O group. Cholesterol reinforces the structure of the cell membrane in animal cells and in an unusual group of cell wall deficient bacteria called the mycoplasmas. The cell membranes of fungi also contain a sterol, called ergosterol. Page 21. The cutaway view of a cell membrane is shown. Figure 1.13. Cutaway view of a membrane with its bilayer of lipids. The primary lipid is phospholipid. However, cholesterol is inserted in some membranes. Access the text alternative for figure 1.13. Page 22. Chemically, a wax is an ester formed between a long-chain alcohol and a saturated fatty acid. The resulting material is typically pliable and soft when warm but hard and water-resistant when cold. Think of a wax candle. Among living things, fur, feathers, fruits, leaves, human skin, and insect exoskeletons are naturally waterproofed with a coating of wax. Bacteria that cause tuberculosis and leprosy produce a wax that repels ordinary laboratory stains and contributes to their disease-causing potential. Proteins, shapers of life. The predominant organic molecules in cells are proteins. To a large extent, the structure, behavior, and unique qualities of each living thing are a consequence of the proteins they contain. The building blocks of proteins are amino acids, which exist in 20 different naturally occurring forms. Table 1.6. Various combinations of these amino acids account for the nearly infinite variety of proteins. Table 1.6. 20 amino acids and their abbreviations. Acid abbreviation characteristic of their R groups. Alanine, arginine, asparagine, aspartic acid, cysteine, glutamic acid, glutamine, glycine, histidine, isoleucine, leucine, lysine, methionine, phenylalanine, proline, serine, threonine, tryptophan, tyrosine, valine, ALA, ARG, ASN, ASP, SICE, GLU, GLN, LE, HIS, EO, LEU, LEAF, MET, FAD, SIR, CHR, TRP, TIR, PAL, nonpolar, plus sign, polar, hyphen, polar, hyphen, polar, polar, plus sign, nonpolar, nonpolar, plus sign, nonpolar, 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 polar, polar, nonpolar, polar, nonpolar. Plus equals positively charged, equals negatively charged. Various terms are used to denote the nature of proteins. Peptide usually refers to a molecule composed of short chains of amino acids, such as a dipeptide, 2 amino acids, a tripeptide, 3, and a tetrapeptide, 4. A polypeptide contains an unspecified number of amino acids, usually more than 20, and is often a small subunit of a protein. A protein is the largest of this class of compounds and usually contains a minimum of 50 amino acids. It is common for the term protein to be used to describe all of these molecules. In Chapter 8, we see that protein synthesis is not just a random connection of amino acids. It is directed by information provided in DNA. Page 23. Protein structure and diversity. The reason that proteins are so varied and specific is that the way they are folded makes all the difference with respect to their function. A protein has a natural tendency to assume more complex levels of organization, called the secondary, tertiary, and quaternary structures. Figure 1.14. The primary, 1 degree, structure is the type, number, and order of amino acids in the chain, which varies extensively from protein to protein. The secondary, 2 degrees, structure arises when various functional groups, called R groups exposed on the outer surface of the molecule interact by forming hydrogen bonds. This interaction causes the amino acid chain to twist into a coiled configuration called the alpha helix, Greek letter alpha helix, or to fold into an accordion pattern called a beta-pleated sheet, Greek letter beta-pleated sheet. Many proteins contain both types of secondary configurations. Proteins at the secondary level undergo a third degree of torsion called the tertiary, 3 degrees, structure created by additional bonds between functional groups. Figure 1.1c. In proteins with the sulfur-containing amino acid cysteine, further tertiary stability is achieved through covalent disulfide bonds between sulfur atoms on 2 different parts of the molecule. Some complex proteins also participate in a quaternary, 4 degrees, structure, in which more than one polypeptide forms a large, multi-unit protein. This is typical of antibodies and some enzymes that act in cell synthesis. Access the text alternative for protein structure and function. Protein structure and function. Figure 1.14 stages in the formation of a functioning protein. Its primary structure is a series of amino acids found in a chain. B. Its secondary structure develops when the chain forms hydrogen bonds that fold it into one of several configurations, such as a Greek letter alpha helix or Greek letter beta pleated sheet. Some proteins have several configurations in the same molecule. A protein's tertiary structure is due to further folding of the molecule into a three-dimensional mass that is stabilized by hydrogen, ionic, and disulfide bonds between functional groups. D. The quaternary structure exists only in proteins that consist of more than one polypeptide chain. Each of the separate chains in this protein has a different color. Protein is a major component of needs. F. And non-copyright by Comstock Images Getty Images. The most important outcome of the various forms of bonding and folding is that each different type of protein develops a unique shape, and its surface displays a distinctive pattern of pockets and notches. As a result, a protein can react only with molecules that complement or fit its particular surface features, like a lock and key. Such a degree of specificity can provide the functional diversity required for many thousands of different cellular activities. For example, enzymes serve as the catalysts for all chemical reactions in cells, and nearly every reaction requires a different enzyme. See Chapter 7. This specificity comes from the architecture of the binding site, which determines which molecules fit it. The same is true of antibodies. Antibodies are complex glycoproteins with specific regions of attachment for bacteria, viruses, and other microorganisms. The functional three-dimensional form of a protein is termed the native state. If it is disrupted by some means, the protein is said to be denatured. Agents such as heat, alcohol, and some disinfectants denature the stabilizing bonds within the chains and cause the molecule to become non-functional, as described in Chapter 9. Page 24. The nucleic acids, a cell computer and its programs. The fourth type of macromolecule nucleic acid, DNA and RNA, are two major representatives of this group. DNA contains a special coded genetic program with detailed and specific instructions for each organism's heredity. It transfers the details of its program to RNA. Helper molecules responsible for carrying out DNA's instructions and translating the DNA program into
What you say? What you say? Read my book. I'm here for sugar. That's why I don't read it. I can hear it. Then I got my PowerPoint presentation.
I need to get a shower. So I track it down.
You got two hands, so you're gonna need to wash back and
No way. I like that is because
that's where I got it from. Myers.
I know you don't have mine either.
So, is that pump right there?
You look at the phone the other day, but I don't
Mom was sorry about that.
She's like, how you gonna value this? I know that house ain't worth that.
We just want to argue.
What she say? Who was she married
Yeah,
to?
they made her house valued at like one something. And she's like, that's gonna make my taxes jump. I was like, okay, that's a good thing. My house ain't worth that. I'm done there. I just listen to her. Say, bro, you just want to buzz. Like, bro,
I'm like, I mean, we value that 300.
hey, so this is a tentative thing. This is for taxes there.
I know. That's good.
But our house is valued at 3 because that's what they keep offering. I was like, bro, you can run up. I said, you get 250 for your house. It ain't worth 250. Oh, you got
You
it.
gotta take your account, man.
But she got it. We'll be working about the participate. I'm not about to fuss and be fine now.
Yeah, your taxes are gonna go up. I ain't recouping that.
I said, Nevada. I said, I'm on it. It's a good thing. No, it's not. My house ain't worth that. Look at that. Did it? It's a choice that we did there. I'm on. It's a choice. You could have been had it done. Marlon does it. I even had David look at it. I'm buying frugal. She frugal. She ain't gonna, if she thinks she gotta spend a dime, she gonna, $40. I see, man. He okay. He okay. Like I said, I see the perfect he's gonna get. Sometimes they're like, calm down, my guy. You didn't need your tablets. Well, she is like, it ain't down in my house at 100 something. It ain't worth that. It's a 2-bedroom outside for you and Northside. That don't mean nothing. I said, yes, it does. The guy goes, she said, they ain't doing no redevelopment. Oh, they're on Kirby all over there. You're getting charged for that shit because all the houses over there cost $250. I know because we were looking. I think that house around the corner went for almost 600 grand, between 500 and 600. Oh, it did? Fine. They redid the house. That house is a row house. The row houses cost money. And I said, you live here. Your area's up. It's a good thing. You got 3 schools in less than 2 miles. 2 of them on your street. Right? She's sitting up there like, my house ain't worth that. You got it, Pimp. I just sat there and was like, honestly, I want my house valued at more. This ain't about living here. This is about what it's worth. Because I can renegotiate, remortgage, refinance, right? I can also, what? Use it as collateral, right? So she was like, my house ain't That's it. If you decided to redo the whole basement, that's damn near an apartment. It's not my fault that you're not maximizing living space.
She's fine.
Ah, man, she's just older in age and fussy.
Cluck.
It is. Whoa! If that's the case, I should have got the Gucci then. Damn. I'm gonna get started with the Ruthie. People said Ruthie to get Gucci. I didn't get no Gucci. So you said, I'm lazy, I'm tired,
I ain't that tired. I said lazy.
No. You said, I am lazy, I'm tired. So,
What do you mean?
guess what? No, you said I'm lazy and tired.
Lazy and sleepy. Lazy and sleepy.
I gave you the roof again, you better, or not, so it was first. No, but first me was, I'm rooty. Look, she's still in the back. You should have ate a third. You ate half.
Tell me, I roofied you. Roofie you over.
Tell me I'm roofie. I roofie you and you didn't even put out.
Wow, me, nigga.
Yes, you did.
That's fine. I'm gonna take it.
No.
You said what?
The nucleic acids, a cell computer and its programs. The fourth type of macromolecule is nucleic acid. DNA and RNA are two major representatives of this group. DNA contains a special coded genetic program with detailed and specific instructions for each organism's heredity. It transfers the details of its program to RNA, helper molecules responsible for carrying out DNA's instructions and translating the DNA program into proteins that can perform life functions. For now, let us briefly consider the structure and some functions of DNA, RNA, and a close relative, adenosine triphosphate, ATP, shown as curly hair, which is the result of particular protein folding patterns. Curly hair is the result of particular protein folding patterns described in Figure 1.14 copyright Ingram Publishing. The double helix of DNA. DNA is a huge molecule formed by two long nucleotide strands linked along their length by hydrogen bonds between nitrogen bases. The pairing of the nitrogen bases occurs according to a predictable pattern. Adenine always pairs with thymine, and cytosine with guanine. The bases are attracted in this way because each pair shares oxygen, nitrogen, and hydrogen atoms exactly positioned to align perfectly for hydrogen bonds. Figure 1.15, page 25. Access the text alternative for DNA structure. DNA structure. A three-part illustration shows the structures of DNA and RNA. Figure 1.15 the general structure of nucleic acid. Access the text alternative for Figure 1.15. Owing to the manner of nucleotide pairing and stacking of the bases, the actual configuration of DNA is a double helix that looks somewhat like a spiral staircase. Just as with proteins, the structure of DNA is intimately related to its function. DNA molecules are usually extremely long. RNA, organizers of protein synthesis. Like DNA, RNA consists of a long chain of nucleotides. However, RNA is usually a single strand, except in some viruses. It contains ribose sugar instead of deoxyribose and uracil instead of thymine. See Table 1.5. Several functional types of RNA are formed using the DNA template through a process called transcription. Three major types of RNA are directly used for protein synthesis. Messenger RNA. mRNA is a copy of a gene, a single functional part of the DNA that provides the order and type of amino acids in a protein. Transfer RNA. tRNA is a carrier that delivers the correct amino acids for protein assembly. And ribosomal RNA, RNA, is a major component of ribosomes. described in Chapter 3. A fourth type of RNA is the RNA that acts to regulate the genes and gene expression. More information on these important processes is presented in Chapter 8. Complex registered sign prep. A nurse is caring for a patient with refractory epilepsy. To enhance medical management, a ketogenic diet has been prescribed. In educating the patient about dietary energy sources, all of the following statements are true, except proteins consist of polymers of nucleic acids. Fat provides more kilocalories per gram than protein. A high-fat diet may cause increased serum cholesterol levels. Starch is an example of a carbohydrate. Answer start revealed content. A revealed content has ended. ATP, the energy molecule of cells. A relative of RNA involved in an entirely different cell activity is adenosine triphosphate. ATP. ATP is a nucleotide containing adenine, ribose, and 3 phosphates rather than just one. Figure 1.16. It belongs to a category of high-energy compounds, also including guanosine triphosphate, GTP, that gives off energy when the bond is broken between the second and third atomar phosphate. The presence of these high-energy bonds makes it possible for ATP to release and store energy for cellular chemical reactions. Breakage of the bond of the terminal phosphate releases energy to do cellular work and also generates adenosine diphosphate, ADP. ADP can be converted back to ATP when the third phosphate is restored, thereby serving as an energy depot. Carriers for oxidation reduction activities, nicotinamide adenine dinucleotide NAD, for instance, are also derivatives of nucleotides. See Chapter 8. A two-part illustration shows the structure of ATP. Figure 1.16 An ATP molecule. The structural formula. Wavy lines connecting the phosphates represent bonds that release large amounts of energy. B. A ball and stick model. Access the text alternative for figure 1.16. Cells, where chemicals come to life. As we proceed in this chemical survey from the level of simple molecules to increasingly complex levels of macromolecules, at some point we cross a line from the realm of lifeless molecules and arrive at the fundamental unit of life called a cell. A cell is indeed a huge aggregate of carbon, hydrogen, oxygen, nitrogen, and many other atoms, and it follows the basic rules of chemistry and physics. But it is much more. The combination of these atoms produces characteristics, reactions, and products that can only be described as living. Page 26. Fundamental characteristics of cells. The bodies of some living things, such as bacteria and protozoa, consist of only a single cell, whereas those of animals and plants contain trillions of cells. Regardless of the organism, all cells have a few common characteristics. They tend to be spherical, ligonal, cubical, or cylindrical, and their protoplasm, internal cell contents, is encased in a cell or cytoplasmic membrane. They have chromosomes containing DNA, and ribosomes for protein synthesis, and they are exceedingly complex in function. Aside from these few similarities, the contents and structure of the three different cell types, bacterial, archaeal, and eukaryotic, differ significantly. Animals, plants, fungi, and protozoa are all made up of eukaryotic cells. Such cells contain a number of complex internal parts called organelles that perform useful functions for the cell involving growth, nutrition, or metabolism. Organelles are distinct cell components that perform specific functions and are enclosed by membranes. Organelles also partition the eukaryotic cell into smaller compartments. The most visible organelle is the nucleus, a roughly ball-shaped mass surrounded by a double membrane that contains the DNA of the cell. Other organelles include the Golgi apparatus, endoplasmic reticulum, vacuoles, and mitochondria. Bacterial and archaeal cells may seem to be the cellular have-nots because, for the sake of comparison, they are described by what they lack. They have no nucleus and generally no other organelles. This apparent simplicity is misleading. However, because the fine structure of these cells is complex, overall, bacterial and archaeal cells can engage in nearly every activity that eukaryotic cells can, and many can function in ways that eukaryotes cannot. Chapter 3 is more delved deeply into the properties of bacterial, archaeal, and eukaryotic cells. Page 27. 1.3 Learning Outcomes. Assess your progress. Name the 4 main families of biochemicals. Provide examples of cell components made from each of the families of biochemicals. Differentiate among primary, secondary, tertiary, and quaternary levels of protein structure. List the 3 components of a nucleotide. Name the 3 nitrogen bases of DNA and RNA. List the 3 components of ATP. Recall 3 characteristics common to all cells. 1.4 Naming, classifying, and identifying microorganisms. The science of classifying living beings is taxonomy. It originated more than 250 years ago when Carl von Linné, also known as Linnaeus, 1701 to 1778, a Swedish botanist, laid down the basic rules for classification and established taxonomic categories, or taxa, singular, taxa. Von Linné realized early on that a system for recognizing and defining the properties of living beings would prevent chaos in scientific studies by providing each organism with a unique name and an exact slot in which to catalog it. This classification would then serve as a means for future identification of that same organism and permit people working in many biological fields to know if they were indeed discussing the same organism. A chicken egg is a single large cell. Copyright Ingram Publishing. The primary concerns of modern taxonomy are still naming, classifying, and identifying. These 3 areas are interrelated and play a vital role in keeping a dynamic inventory of the extensive array of living and extinct beings. In general, nomenclature, naming, is the assignment of scientific names to the various taxonomic categories and to individual organisms. Classification is the orderly arrangement of organisms into a hierarchy. Identification is the process of discovering and recording the traits of organisms so that they may be recognized or named and then classified. Nomenclature. Many macroorganisms are known by a common name suggested by their dominant features. For example, a bird species may be called a red-headed blackbird or a flowering plant species a black-eyed Susan. Some species of microorganisms are also called by informal names, including human pathogens such as Gonococcus, Neisseria gonorrhoeae, or fermenters such as brewer's yeast, Saccharomyces cerevisiae, or the recent Ironcitobacter, Acinetobacter baumannii. But this is not the usual practice. If we were to adopt common names such as the little yellow coccus, the terminology would become even more cumbersome and challenging than scientific names. The method of assigning a scientific or specific name is called the binomial, to-name, system of nomenclature. The scientific name is always a combination of the genus name followed by the species name. The genus part of the scientific name is capitalized, and the species part begins with a lowercase letter. Both should be italicized, or underlined if using handwriting, as follows. The handwritten text specialty is italicized and underlined. Page 28. The tree hopper and its discoverer, Sylvie, below. Sylvie, at the age of 5, with her mother the biologist Dr. Laura Sullivan Beckers. Above copyright Robin Pizzo. Top, copyright Dr. Laura Sullivan Beckers. Bottom. The 2-part name of an organism is sometimes abbreviated to save space, as in E. coli, but only if the genus name has already been stated. The inspiration for names is extremely varied and often rather imaginative. A biology professor from Murray State University in Kentucky was planting flowers in her garden with her 2-year-old daughter Sylvie, when Sylvie discovered something. It was a species of treehopper insect that her mother was not familiar with. It turns out that it was an as yet undiscovered species, and Sylvie's mom, Dr. Laura Sullivan Beckers, named it in honor of its discoverer, her daughter. Its binomial name is now Hebetica sylviae. Some species of microbes have also been named in honor of a microbiologist who originally discovered the microbe or who has made outstanding contributions to the field. Other names may designate a characteristic of the microbe, shape, color, a location where it was found, or a disease it causes. 2 examples of specific names, their pronunciations, and their origins are Staphylococcus aureus, Staphylococcus aureus, Gr. staphyl, bunch of grapes, coccus, berry, and Gr. aureus, golden. The species looks like a bunch of grapes under a microscope, and its colonies are golden yellow on agar. It is a common bacterial pathogen of humans. Lactobacillus San Francisco, Lactobacillus San Francisco, L-lacto, milk, and Bacillus, little rod, a bacterial species used to make sourdough bread, for which San Francisco is known. Here's a helpful hint. These names may seem difficult to pronounce and the temptation is to simply slur over them. But when you encounter the names of microorganisms in the chapters ahead, it will be extremely useful to take the time to sound them out and repeat them until they seem familiar. Even experienced scientists stumble the first few times through new names. Stumbling out loud is a great way to figure them out. and you are much more likely to remember them that way. They are less likely to end up in a tangled heap with all of the new language you will be learning. Classification. Classification schemes are organized into several descending ranks, beginning with the most general all-inclusive taxonomic category and ending with the smallest and most specific category. This means that all members of the highest category share only one or a few general characteristics, whereas members of the lowest category are essentially the same kind of organism. That is, they share the majority of their characteristics. The taxonomic categories from top to bottom are domain, kingdom, phylum or division, class, order, family, genus, and species. That means that each kingdom can be subdivided into a series of phyla or divisions. Each phylum is made up of several classes. Each class contains several orders, and so on. Because taxonomic schemes are to some extent artificial, certain groups of organisms may not exactly fit into the main categories. In such a case, additional taxonomic levels can be imposed above, super, or below, sub, a taxon, giving us such categories as superphylum and subclass. Let's compare the taxonomic breakdowns of a human and a protozoan, protozoan, to illustrate the fine points of this system. Figure 1.17. Humans and protozoa are both organisms with nucleated cells, eukaryotes. Therefore, they are in the same domain, eukarya, but they are in different kingdoms. Humans are multicellular animals, kingdom Animalia, whereas protozoa are single-cellular organisms that, together with algae, belong to the kingdom Protista. To emphasize just how broad the category kingdom is, ponder the fact that we humans belong to the same kingdom as jellyfish. Of the several phylum within this kingdom, humans belong to the phylum Chordata, but even a phylum is rather all-inclusive, considering that humans share it with other vertebrates as well as with creatures called sea squirts. The next level, class Mammalia, narrows the field considerably by grouping only those vertebrates that have hair and suckle their young. Humans belong to the order Primates, a group that also includes apes, monkeys, and lemurs. Next comes the family Hominoidea, containing only humans and apes. The final levels are our genus, Homo, all modern and ancient humans, and our species, Sapiens, meaning wise. Notice that for the human as well as the protozoan, the taxonomic categories in descending order become less inclusive and the individual members more closely related. In this text, we are usually concerned with only the most general, domain, kingdom, phylum, and specific, genus, species, taxonomic levels. Phylogenetic tree, page 29. An illustration shows the taxonomic classification of Homo sapiens and Paramecium caudinum. Figure 1.17 sample taxonomy. Two organisms belonging to the Eukarya domain, traced through their taxonomic series. On the left, modern humans, Homo sapiens. On the right, a common protozoan, Paramecium caudinum. Access the text alternative for figure 1.17. Page 30. Identification. Discovering the identity of microbes we find in the environment or in diseases is an art and a science. The methods used in this process are extensively described in Chapter 2 and in Chapter 15. The origin and evolution of microorganisms. As we indicated earlier, taxonomy, the science of classification of biological species, is used to organize all of the forms of modern and extinct life. In biology today, there are different methods for deciding on taxonomic categories, but they all rely on the degree of relatedness among organisms. The scheme that represents the natural relatedness, relation by descent, between groups of living beings is called their phylogeny, gr phylum, race or class, genesis, origin or beginning. Biologists use phylogenetic relationships to refine the system of taxonomy. To understand the natural history of and the relatedness among organisms, we must understand some fundamentals of the process of evolution. Evolution is an important theme that underlies all of biology, including the biology of microorganisms. As we said earlier, evolution states that the hereditary information in living beings changes gradually through time and that these changes result in various structural and functional changes through many generations. The process of evolution is selective in that those changes that most favor the survival and reproduction of a particular organism or group of organisms tend to be retained, whereas those that are less beneficial to survival tend to be lost. This is not always the case, but it often is. Charles Darwin called this process natural selection. COVID-19. During the COVID-19 pandemic, it was declared that the virus had evolved from being able to infect non-human animals to being able to infect humans. This is an example of small, random changes in the genetic information that happen to provide the virus a new skill, the ability to infect a new species. Usually, evolution progresses toward greater complexity, but there are many examples of evolution toward lesser complexity. This is called reductive evolution. This is because individual organisms almost never evolve in isolation but as populations of organisms in their specific environments, which exert the functional pressures of selection. Because of the nature of the evolutionary process, the phylogeny, or relatedness by descent, of organisms is often represented by a diagram of a tree. The trunk of the tree represents the origin of ancestral lines, and the branches show offshoots into specialized groups of organisms. This sort of arrangement places taxonomic groups with less divergence, less change in the heritable information, from the common ancestor closer to the root of the tree, and taxa with lots of divergence closer to the top. A universal web of life. The first trees of life were constructed a long time ago on the basis of just two kingdoms, plants and animals, by Charles Darwin and Ernst Haeckel. These trees were chiefly based on visible morphological, shape characteristics. It became clear that certain microorganisms such as algae and protozoa, which only existed as single cells, did not truly fit either of those categories. So a third kingdom was added. It was named Protista, or protozoa. Eventually, when significant differences became evident among even the unicellular organisms, a fourth kingdom was established in the 1870s by Haeckel and named Manera. Almost a century before Robert Whittaker extended his work and added a fifth kingdom for fungi during the period of 1959 to 1969. The relationships that were used in Whittaker's tree were those based on structural similarities and differences, and the way these organisms obtained their nutrition. These criteria indicated that there were 5 major taxonomic units, or kingdoms: the Manera, protists, plants, fungi, and animals. Each organism in these 5 categories consisted of one of the 2 cell types, those cells lacking a nucleus and the eukaryotic cells. Whittaker's 5-kingdom system quickly became the standard. Shown as a handful of soil, it is the home to thousands of different kinds of organisms. A handful of soil is home to thousands of different kinds of organisms, including a wide diversity of fungi, bacteria, viruses, and protozoa. Copyright Pixel H. The Top Stock. Page 31. With the rise of genetics as a molecular science, newer methods for determining phylogeny have led to the development of a differently shaped tree, with important implications for our understanding of evolutionary relatedness. Molecular genetics allowed an in-depth study of the structure and function of the genetic material at the molecular level. In 1975, Carl Weiss discovered that one particular macromolecule, the ribonucleic acid in the small subunit of the ribosome, abbreviated SSU-RNA, or 16S-RNA, was highly conserved, meaning that it was nearly identical in organisms within the smallest taxonomic category, the species. Based on a vast amount of experimental data, Weiss hypothesized that 16S-RNA provides a biological chronometer or a living record of the evolutionary history of a given organism. Extended analysis of this molecule in prokaryotic and eukaryotic cells indicated that all members in a certain group of bacteria, then known as archaebacteria, had 16S-RNA with a sequence that was significantly different from the 16S-RNA found in other bacteria and in eukaryotes. This discovery led Carl Weiss and collaborator George Fox to propose a separate taxonomic unit for the archaebacteria, which they named Archaea. Under the microscope, they resembled the structure of bacteria, but molecular biology has revealed that the Archaea, though seemingly bacterial in nature, were actually more closely related to eukaryotic cells. To reflect these relationships, Weiss and Fox proposed an entirely new system that assigned all known organisms to one of the 3 major taxonomic units, the domains, each being a different type of cell. Turn to the beginning of this chapter and re-examine Figure 1.1, which is a depiction of the 3-domain system. The domains are the highest level in hierarchy and can contain many kingdoms and superkingdoms. Cell types lacking a nucleus are represented by the domains Archaea and Bacteria, whereas eukaryotes are all placed in the domain Eukarya. Analysis of the 16S-RNAs from all organisms in these 3 domains suggests that all modern and extinct organisms on Earth arose from a common ancestor. Therefore, eukaryotes did not emerge from bacteria and Archaea. Instead, it appears that bacteria, Archaea, and eukaryotes all emerged separately from a different, now extinct, cell type. To add another level of complexity, the most current data suggests that trees of life do not truly represent the relatedness of organisms in their totality. It has become obvious that genes travel horizontally, meaning from one species to another in non-reproductive ways, and that the tidy generation-to-generation changes are complicated by neighbor-to-neighbor exchanges of DNA. For example, it is estimated that 40% to 50% of human DNA has been carried to humans from other species by viruses. For these reasons, most scientists like to think of the web as the proper representation of life these days. Nevertheless, this new scheme does not greatly affect our presentation of most microbes because we will discuss them at the genus or species level. Keep in mind that our methods of classification or evolutionary schemes reflect our current understanding and will change as new information is uncovered. Please note that viruses and prions are not included in any of the classification or evolutionary schemes because they are not cells or organisms. Their special taxonomy is discussed in Chapter 5. 1.4 Learning Outcomes. Assess your progress. Differentiate among the terms nomenclature, taxonomy, and classification. Create a mnemonic device for remembering the taxonomic categories. Correctly write the binomial name for a microorganism. Draw a diagram of the three major domains. Explain the difference between traditional and molecular approaches to taxonomy. Page 32. End of chapter content. Case file wrap-up. If you have a bacterial infection, your doctor is likely, but not in all cases, to prescribe an antibiotic. Antibiotics are drugs that are designed to harm microbes but not harm the human host. That is their specific job, to target the microorganism. So if you have an illness that is not caused by a microorganism, you should not take antibiotics. Copyright Michael Williams. Photo of Kelly Cowan with student. Science Photo Library RF Getty Images. White blood cell. If you decide to go into healthcare as a profession, you will see a few common infections very frequently, but there will also be a wide variety of infections that you will likely only encounter once or a few times in your career. No one expects you to remember everything about every possible infection you studied here. What's important is that you become familiar with important patterns of disease and the ways that our body, and the treatments we apply, affect them. Page 33. The microbiome. Meet your microbiome. In the past few years, a new word has popped up on news feeds and websites. Microbiome. It refers to the sum total of all the microbes in a certain environment. The human microbiome consists of all the viruses, bacteria, fungi, and protozoa that call the human body home. Trillions of these microorganisms live on our body, as part of our natural biology. Unless something goes wrong, they do not cause disease, and, in fact, are necessary parts of human development and ongoing life. An illustration of human body shows the digestive system highlighted. Structures of bacteria, viruses, fungi, and protozoa are also shown. Copyright Anna Smirnova Alami Stock Vector. The Human Microbiome Project. HMP began in 2008, using techniques to identify body microbes that did not require growing the microbes separately in the lab, a technique scientists have relied on since the mid-1800s, but instead identified them on the basis of their genetic material. The HMP produced a staggering array of results, and they keep coming at breakneck pace. We have learned that the microbiome may differ whether you were delivered via cesarean or vaginal birth. We have learned that the gut microbiome, the microbes living in your intestinal tract, may influence not just your intestinal health but also your likelihood to experience autoimmune disease, your weight, and even your mood. We know how the composition of the microbiome of different body systems, your skin, your eyes, your lungs, differs in health and in disease. In short, we have learned that the characteristics of your microbiome determine your own human biology and what types of experiences you will have as an organism. But there is a big note of caution here. The explosion of information about the human microbiome has led to some very preliminary science, and a lot of careless journalism. One leading microbiome researcher even calls it microbiomania. Many journalists gloss over the difference between correlation. These two things were observed at the same time, and causality. This thing led to this other thing. So we get reporting that suggests that elite cyclists should be poop doping, getting transplanted fecal material that contains the donor's gut microbiome, to improve their performance, when there is no real evidence for it. Right here at the end of every chapter in this book, We will tell a short story about the microbiome as it pertains to the subject matter in the chapter, and try to address the quality of the science and the reporting behind the story. Page 34. Introduction to microbes and their building blocks. 1.1 Microbes. Tiny but mighty. Microorganisms in this book are archaea, bacteria, fungi, protozoa, helminths, viruses, and prions. Some are cellular and some are not cells. Microbes are everywhere on the planet and are critical for nutrient and energy cycling. CTCTR. Lucille K. Gaylord. 1.2 Microbes. Website. And nucleic acids. Macromolecules are generally known as polymerization of smaller molecular subunits. Classifying and identifying microorganisms. Classifying and identifying microorganisms is called taxonomy. All cellular organisms belong to one of three domains: eukaryote, bacteria, archaea. Page 35. Smart Grid. From knowledge to critical thinking. This 21 question grid takes the topics from this chapter and arranges them with respect to their
Downstairs, there's a and oh, I'm looking.
Hello again. I'm keeping a close watch. You have a heart of gold. Don't let them take it from you. Hello again. I'm keeping a close watch.
Girl, guess what my phone said? We're sorry. Nothing you took care of. That's all the restrictions. I was thinking about my
You have a heart of gold. Hello again. I'm keeping a close watch.
I like a light. But I got 8 years done the best I ever got done.
You have a heart of gold. Don't let them take it from you.
Perfectly fair, like,